Hi everyone. In this lecture, we'll continue with transcription in bacteria, that is initiation of transcription, and we'll also touch upon the elongation of transcription in E. coli. So this is a diagram where we'll see how transcription is initiated in E. coli. This is the gene, and the RNA coding sequence is shown in pink color. And the upstream of it, the sequence shown in green is a promoter sequence. And this is the yellow color thingy is the RNA polymerase holoenzyme. This part is the RNA polymerase core enzyme having the alpha, alpha, beta, beta prime, and omega subunits. It's a multi-subunit enzyme as we have seen in the previous lecture. And this is the sigma factor. Sigma factor along with the core enzyme makes this as an holoenzyme. So this RNA polymerase holoenzyme binds at the promoter sequence. And the way RNA polymerase interacts with the promoter sequence dictates to the enzyme the direction of transcription, as we have seen in the previous lecture, which strand is a template strand, and where exactly to begin the transcription. And the RNA polymerase holoenzyme orients itself at the promoter, so the transcription of the gene occurs at the correct site every time the gene is transcribed. And we have seen in the previous lecture how holoenzyme is involved in the initiation of transcription, and the difference between the holoenzyme and core enzyme is the sigma factor, and sigma factor plays a key role in recognizing the key sequences in the promoter. So in this first step, the RNA polymerase holoenzyme recognizes the promoter at the -35 region. The -35 region is the -35 consensus sequence, TT-GACA. And after the RNA polymerase has recognized the -35 region and it binds to the full promoter, the denaturation has not happened yet. And this is the reason this stage is called as the close promoter complex. So the RNA polymerase has recognized the -35 sequence. It's bound to the full promoter, but denaturation has not happened yet. So close promoter complex. As the initiation continues in the next step, RNA polymerase binds more tightly in the -10 region, the -10 consensus sequence, the Pribno box or the Tata-At box, and untwisting of the DNA happens, that is the denaturation occurs. Denaturation means by breaking of the hydrogen bonds between the nitrogen bases, and this denaturation leads to the opening of the DNA called as transcription bubble, which we can see here. And at this point, this untwisting occurs in the -10 region, and at this point, RNA polymerase is correctly oriented to begin the transcription at the +1. This is a +1. +1 is the first nucleotide to be copied into mRNA. And this mRNA is being synthesized now. So the +1 begins from where the RNA coding sequence, the sequence shown in pink, begins. And this part is the first nucleotide of the mRNA. There are many sigma factors, not just one. This sigma factor, which is in the picture, is the sigma 70. It's called as sigma 70 because it has a molecular weight of 70,000 daltons. And this sigma 70 is a housekeeping sigma factor, which recognizes the -10 and -35 consensus sequences present in most of the promoters in bacteria. And this is involved in the transcription of most of the genes. But as I said, there are other sigma factors, and under special circumstances, those sigma factors associate with the core RNA polymerase instead of sigma 70, and then the holoenzyme with those sigma factors can recognize special promoters required for the transcription of those genes under those conditions. So Once the denaturation happened and the mRNA synthesis begins, let's see what happens next. This, by the way, this step is called as the open promoter complex because the denaturation has happened. In the next step, after the 8 to 9 nucleotides have been polymerized. So in this step, you will see that the mRNA synthesis has begun, and after 8 to 9 nucleotides have been polymerized, sigma factor dissociates from the core enzyme. So sigma factor released, and sigma factor leaves the core enzyme because sigma factor is not required for the elongation and termination stages of transcription. We have seen this in the previous lecture as well. Sigma factor is involved in initiation of transcription because without sigma factor, the core enzyme can do the transcription, but it can start from anywhere, resulting in incorrect mRNA, and then it won't be functional. So sigma factor helps the RNA polymerase recognize those -10 and -35 sequences, and it reduces the non-specific binding of the RNA polymerase. It helps RNA polymerase binds only at promoters. And since now the RNA polymerase is bound to the promoter because of sigma factor, the untwisting has happened, mRNA synthesis has begun. Now sigma factor is no longer required because we are into the elongation. And sigma factor was involved or required only for initiation. Sigma factor will leave and rest of the transcription would be carried out by core enzyme alone. And the RNA polymerase elongates the new RNA chain. The enzyme untwists the DNA ahead of it. So in this step, the untwisting has happened till here. In the next step, further untwisting has happened. So the enzyme untwists the DNA ahead of it, keeping a transcription bubble about 25 base pairs. And behind the untwisted region, the DNA can reform into the double-stranded helix. And about 9 bases of the new RNA are bound to the single-stranded DNA bubble. So around 9 bases of the RNA are bound to the single-stranded bubble, while the remainder of the RNA exit the enzyme in the single-stranded form. So this was the elongation of transcription in E. coli. In this lecture, in this video, we have continued with the initiation of transcription in E. coli from the previous lecture. And we have seen with the help of this diagram how the sigma factor helps the RNA polymerase enzyme to make direct contact with the -35 and -10 consensus sequences. And what is closed promoter complex, what is open promoter complex, and how once the RNA is synthesized or the synthesis begins, the sigma factor leaves and rest of the transcription is carried out by the core enzyme alone. And during the elongation, how the enzyme untwists the DNA ahead of it, but behind it, the DNA reforms the double-stranded helix, and some of the RNA exits the enzyme in the single-stranded form, while some is part of the single-stranded DNA bubble.